now let's get into translation. Right? So translation is converting the language of nucleotides into the language of amino acids. Meaning, you're reading on your mRNA, it says it needs eggs. Well, who do I call that has the eggs? Who's the phone number that I call for the brownie mix? Who's the one for the vanilla extract? You see what I'm saying? So that's where this code is, the telephone book, the address book. We have to introduce you to this genetic code, okay? So this isn't the actual translation, this is actually who do you call, okay? So that's where that is. And this happens in the ribosomes. I make the phone is actually in my kitchen, okay? And it's going to translate the sequence of nucleotides into the sequence of amino acids. And the ribosomes, as you've seen before, MUO is made up of two units, two subunits, a 40X and a 60X. And they actually kind of clam together. So when this mRNA comes into the cytoplasm, and the ribosome knows exactly where to clam up on that mRNA, where it begins to do this translation. Okay? There's 3 sites that are very prudent for you guys to understand. Okay? There's something called the A site, the P site, and the E site. And it's pretty easy to memorize because it says 8. Okay? What's going to happen first is that once we figure out, I'm going to show you the genetic code, once we figure out who to call, and they're our tRNA, they're going to be carrying the amino acid. Mom is carrying the egg is the amino acid. Once we know who to call, then what's going to happen is Mom is going to drive up on the driveway in the A spot, okay, and since no one's over there, she could drive up to the P spot. If someone was there, you got to wait in line. Okay? And then my garage is where you can go right through the house. Okay? The garage would go right through the house. So you're going to go from the A spot, which is the waiting area. The P spot is where you're actually going to come out of the car and put that amino acid into the bowl. And the E site is where you're going to drive up to go back into your car, you're going to go through the garage, and you're going to leave that way. Okay? So it's a point where you have the amino acid, where the amino acid is in the transition of coming off, and then you have a spot on the ribosome where there is no amino acid, and then you can drive off to wherever. Okay? So there's 3 spots on here. Okay? Now, I'm going to show you that on a picture. So let's talk about this genetic code. How do you know whose phone number to call carrying these specific amino acids? So the system, and don't ask how they came up with this, they did. And it's the same system for birds, humans, yeast, dinosaurs, whatever. It's the same genetic code. Okay? I don't know who came up with it. Someone's got some words to guess. People who have no social life, I know that. Right? So we just accept that this is what it is, okay? You don't have to memorize anything, just how to go about reading it, because you'll be utilizing it, okay? I'll give you the genetic code on the test that you can use. So they work by what we call base triplets, the sequence of 3 DNA nucleotides that stand for a certain amino acid, AA amino acid, okay? You also have a codon, and that's where this comes in, okay? I kind of drew you here. This is a base triplet that's on the DNA. What we're doing over here is a codon. So the codon kind of came from the base triplet of the DNA. So you have this 3-base sequence, and keep in mind that I only did three little bases there, but this is usually, it could be, let's say, 1,000 bases. Okay? And there's these 3 base sequences in the mRNA, and we call them codons. The people who came up with this found out that there's 64 possible codons to actually code for 20 amino acids. Doesn't make sense, right? Well, there's certain codons, there might be, let's say, 6 codons that actually code for the same amino acid. I'll show you what that means by that. We also have codons that don't just code for amino acids. 61 do that, but then we have 3 other ones that don't code for amino acids, but they code for instructions. Instruction to say, this is where we're going to begin. All right, on your recipe, it'll say number 1. That tells you that's where it begins. And then there's going to be a couple that will actually signal for stop. All right, so you got these over here that code for stop. Meaning, when you see on the recipe it says, enjoy. That tells me my recipe's done. Right? There's something that does that. So these numbers don't add up. If it's 61 out of 64 that code for amino acids, there should be only 3. But I'm seeing 1, 2, 3, 4. That's because the start codon, which is an instruction, also codes for an amino acid called methionine. So that's how you explain the number issue. Okay? All right. So this is the genetic code. I've seen a few different ways that people actually use it. This, to me, is the simplest way that you can see it. I've seen it where there's a circle. I don't know what you've had in high school, but this is just as easy. Let's say we have a codon is something like AUU. Okay? So this is what we got to do. We have to figure out AUU codes for which amino acid. That phone number is going to code for a certain person to bring eggs. That's the code. So how do you read this? Pretty simple. First base, second base, third base. So on the first base, you're looking over here in the purple area here, and you're looking for A. So therefore, here's where it is. Okay? So you know that the first base is going to be somewhere in this column over here. Okay? The second base is U, which is going to be here. So you know that if it's AU, it's got to be in this area right here. And the third base, which is U, is going to be here, and you'll see it here. So there's AUU. And that's going to code for this amino acid abbreviated by 3 letters. Isoleucine? Yeah, isoleucine. All right? You won't have to know it. You just have to recognize and maybe write down the 3-letter abbreviation. I mean, the other way you could do this is say, oh, AUU. Let's go through this. Where's AUU? It's going to be all this stuff. There it is. But if you want to waste time and do that, go right ahead. All right? Because there'll be a few of those on your exam, and you want to do it fast. Okay? So if I said this codon, CUA, don't say what it is, just look at that, give it a moment. L-E-U, right? Which is leucine, right? So you're going to go, so C, U, A, right there, C-U-A, and that's going to code for leucine, okay? Let's do one more, okay? Much easier than Sudoku, trust me. Let's do, I don't know, let's do G-A-G, gag. What do you think? Yeah, glucine, right over here. So G, A, G. So that way, it brings it right over here. Gag. And it's going to code for glucine. All right? Sometimes it'll also, see here's the A and G for methionine, or the start one, but then there's also 3 stop instructions up there too, coding for those. Okay? Questions about that? That's not bad, right? You can handle that. Alright? But don't get upset if you see this genetic code on the exam and you're just thinking WTF. I mean, this is the easy part. Okay? Alright, so now you know how to do the genetic code. Alright, now here's the translation steps. Okay? You can read this on your own. It's just easier for me to just show you the picture so you can see what goes on. Okay? This whole thing, well, actually, I should do a little bit of it. Alright, so here, the translation steps. So the mRNA is going to attach to the small subunit of the ribosome, right? The 40S subunit. And the tRNA, all right, that's going to code for that, is going to come to the A spot on the ribosome. All right? Now, the mRNA has a codon on it. The tRNA has what we call an anticodon. So if I had, it's the complementary base. So if I had, let's say, if the mRNA has the codon of GAG, we'll use the same one, then the tRNA that's going to complement that is going to be what? Right, CUC. So that's going to be on the tRNA, and then so there, you see the code that's happening over here. There's the recipe, but we're really going to get the amino acid for it. That's how it is. No, this is calling for eggs. So now I can call mom, who has this anticodon, who has the egg. So it's confusing because everything's in threes. You got base triplets from the DNA, you've got the codon that's on the mRNA, and you've got the anticodon that's on the tRNA. Don't get mixed up with all those things. They're all in threes. Okay? So what will happen is, this is the first tRNA that's going to go to the A spot. Okay? And first tRNA will always be a start instruction, which happens to also code for methionine. Okay? That's probably the only amino acid you really do need to know is methionine, for at least the test here. You could go into genetics and learn more about this kind of stuff in other courses, but we don't need you to memorize all the different amino acids. Just understand what is an amino acid, and these names you can just recognize, but I won't ask you about those, except for methionine. Okay? So the tRNA, once it goes to the A spot, can move over to the P spot of the, or P site of the ribosome. And this is where the tRNA is now going to be coupled up with the, well actually this is where the amino acid is going to be added, but this is the first one, so there's nothing to add to it. A peptide bond connecting the amino acids as we keep on putting all these ingredients in the bowls. And these covalent bonds, well, the peptide bond is a covalent bond, right? It's a certain type. Peptide bonds connect amino acids together to give you proteins. Okay? And then the first tRNA moves, once the amino acid comes off, then what's going to happen is, in the process, the amino acid goes off the tRNA into the E spot, or when it moves to the E spot. I got the video, it'll show you a little bit better. All right? And while that's in the E spot, another tRNA will be coming in from the A and going to the P spot and so forth. And the third one comes on and so forth. Until a stop comes on. A stop codon. So what's going to happen here is this. All right? Here's the mRNA. Okay? It's a strand. It's a single-stranded thing. And you have codons on the mRNA. A tRNA kind of looks weird, different, but it's still one strand. But it kind of comes together like this. It's being held, it's in this format and being held by hydrogen bonds. Okay? Certain bases on here actually are complementary on each other, and they're held by hydrogen bonds. And it makes this structure. So you've got this tRNA that's over here. The anticodon is here that would actually correspond to the codon. We've got 3 base pairs over here. And at the other end, there's going to be the amino acid that's going to come off of that and be attached to the previous amino acid that this protein is being made from. So, what's going on here is this. Here you have, all right, let me orient you. So here's the ribosome, 2 parts put it together. This is the mRNA coming through here with all the codons. It'll first go to, there's going to be a codon that first goes to the A site, and that's where this tRNA, which has the anticodon here, is GAA, and the amino acid is over there, in this case it's leucine. This tRNA will go to the A site because that is corresponding. GAA goes with CUU. Perfect. Then this is going to, this tRNA, once it's bound, is going to go to the P site, and we're going to start putting that amino acid and attaching it to the one previous over here. And now that this one, which is now over here, goes to the E site, and then it could break off without the amino acid. The amino acid will get attached later on through the cytoplasm to be done again. So we could use this same tRNA again. Meanwhile, another tRNA comes on, whatever this is over here, goes through the process, and that amino acid gets added to this. And meanwhile, you get this long chain that's happening here. I'm putting the stuff in the bowl to make the brownie. All right? Showing the same thing. All right, here's your ribosome, there's your mRNA, and in this case it's coming this way though. So here's your tRNA, it kind of goes here, moves over a little bit, this amino acid gets attached to that, and then this tRNA can break off without the amino acid over here. And the next one comes on, next one, next one. It's an assembly line. Okay? Just showing the same thing, where it goes step by step. But what they did over here makes it a little bit easier to visualize, is that they're putting numbers instead of the names of the amino acids. So you can actually see where they're going, right? So the first one goes on, this is amino acid number 1, here's amino acid number 2 coming over here, amino acid number 2 and 1's over here, then you got number 3, and amino acid 1 gets attached to 2 over here, but then the transfer RNA comes off from the first one, and it goes on and on and on. get this whole thing. Once you see it, you'll see. It's not, and I got a little video to show you too. Okay? Again, showing the same thing, but in this case, if you wanted to, you could go step by step and see it over here. It gets a little bit confusing than what you learned in high school. You have more steps to it. And of course, we can't just do one protein at a time. We've got to use that kitchen or other kitchens. You know, if there's going to be a bake sale, well, we've got to have a lot of kitchens making a lot of different things. Not just brownies, cupcakes, pizza, whatever. I don't know. But I'm just saying, you've got to have a lot of proteins being made all at the same time. Thousands of proteins being made. And that's what's happening here. All right? What's happening is that, you know, you've got the mRNA that's happening over here. There's ribosomes here, and you start seeing where the proteins in green are being built. There's thousands of them happening at the same time. Okay? So you have different proteins happening. Again, what this is trying to show you the difference between transcription and translation. You see, transcription happens in the library, in the nucleus. Whereas translation happens outside the library. It's going to happen on the ribosomes in the cytoplasm. So you're seeing where they're actually going to be taking place. Okay? If you think about it, don't answer. Just think about it. If the tRNA has an anticodon of that, then what would be the codon of mRNA? This is A, B, C, D, E. Right? A, G. Okay? Not too bad, right? Baby steps. You'll get it. All right, now, let me show you one video I want to show you is a translation, and then the second video I want to show you the whole thing. Okay? They're short. They're like 2 minutes each. Okay?
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Let me show you this last one. This is the whole thing now, proteins. This one I like the sound of it.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traveling in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.